I would say Joe is probably widely recognized as one of the leading figures in our generation as a cell biologist. He was in love with cell biology. He was in love with microscopy. And it was that enthusiasm for science that rubbed off on his trainees as well.
So Joe would take us out on these nature walks and demonstrate to us in live motion his curiosity about everything.
What were the birds that we were seeing? Were we seeing any insects? He was somebody that you could get along with and would be just as comfortable talking to somebody sitting on a train, finding out about them as he was talking to somebody at the Nobel Prize ceremony. He got interested in books, historical, natural history. He'd always talked very warmly about his mother and how she fostered his interest. By the 5th grade, he was raising his own frogs from the pond that were in his yard and taking photographs of it and making a book that he could give to his teacher that illustrated just the same things that he did for the rest of his life.
So he became a naturalist and had a deep understanding of the unique properties of different animal model systems. It was his lifelong dream to try and visualize the position of genes on chromosomes. People had mapped where genes were on chromosomes, but seeing is believing, and so he really wanted to be able to identify by looking at chromosomes through a microscope where a particular gene was located.
In modern terms of understanding how our DNA works, we are more and more aware that we have all these letters in our genome. When you think about trying to understand any short segment of that, the short segments comprise your genes. All these pieces mean something, but how can you study that? How can you light it up, literally make it light up? The Where's Waldo analogy is that the view that you have when you spread out chromosomes is that crazy picture. There's so many people, and I think that represents all the genes. Waldo is the gene of interest. My strategy when I play Where's Waldo, though, is to literally look line by line till I find it. That's very, very cumbersome and time limited. So you want a quick way to sort of snapshot it. So by having a probe that matches that target, you can try and just wash it over, and this probe is labeled, and you can hit that target. Suddenly, when you hit the button to look at just the label, out of all of that mess, you finally see Waldo.
Joe's very first funding was from the National Science Foundation. Years later, those findings and that wealth of knowledge about mechanisms and how things work can be invaluable for developing vaccines or new tools for diagnosis and so forth. This is used for diagnostic purposes as well as to try and understand how far cancer has advanced. One that's near and dear to my heart, because in 2006 I had breast cancer, was to see if there are extra copies of a cancer-causing gene called HER2, which previously would have been a death sentence. With the launch of immunotherapy, you can target specifically the cancer cells, and so the patient is treated with the antibody and now has actually a better prognosis.
So the applications for in situ hybridization, it's really useful to consider the context. Joe developed this out of pure curiosity.
Joe wasn't really looking ahead to those medical advances. The method itself and the development of the method was something that was going to be useful to the whole field of scientists. At the time that I was a graduate student, it was expected that if you got a PhD as a woman in science, you would end up for your career being a research associate in a lab run by a man. But Joe had other expectations for us, and that is that we would have our own independent careers as successful scientists. Somebody noted how many students were women from Joe's lab and named us the Gall girls, and so that name kind of stuck.
He did support women in science, but I think it was maybe incidental to the fact that he was just a brilliant mentor.
In fact, the women that he trained went on, many of them, to amazing careers, including 4 members of the National Academy of Sciences, president of the Genetic Society of America, and of course, a Nobel laureate as well. The federal government is so important in terms of being able to support basic biomedical research so that we can get a better understanding and growth of knowledge of basic mechanisms of how things work, because piecing that all together down the pike can lead to significant medical advances.
a young researcher could learn to follow their passion and not be swayed necessarily by what other people are doing, to think on your own. There's still lots of problems to be solved and questions to be asked.
Go into this as a profession if you really truly enjoy the excitement of exploration of the unknown, of how things work in the natural world. That's the nature of humankind, and that's how we've advanced to where we are today.
I think my dad hoped that in his scientific career he would do something useful. Doesn't everybody start in a laboratory thinking, I'm going to discover a cure for cancer? But he actually did. And that was probably somewhat unexpected.
It wasn't a goofy idea, but it certainly didn't have anything to do with cancer.
Dr. Rosenberg was very personable, very helpful to his students, and had good communication skills with everyone. He was a brilliant biophysicist, but not a chemist.
But that's actually maybe one reason why this worked for him. Innovation often comes from the side. He would sit for hours in the living room and just listen to classical music. He would definitely use that as a way to think through things.
This whole story began when he and Dr. Loretta Van Camp were doing experiments in bacteria. They wondered if passing electric current through it would change the bacteria in some way. They did a bunch of experiments which demonstrated that when that happened, it was a platinum electrode that was causing this to happen.
It was a very slow process over approximately 5 years. But he was able to put together all the pieces to realize that it was a platinum compound that did it.
We wonder if it might do the same thing to cancer cells.
He contacted the National Cancer Institute to ask them for support in testing these compounds against tumors in mice. The National Cancer Institute denied him that request initially. He knew there was value in it, and he got a group of students and postdocs together, and I was his first postdoc, to begin studying the whole process and prepare a pure drug that could be used in treating mice. Work over many, many years that finally led to approval in 1978 of cisplatin. And of course, you know what results were after that has been an amazing drug still in the clinic after basically 47 years.
Cancer at a cellular level is actually pretty easy to understand. It basically is the phenomenon of uncontrolled growth. Cisplatin probably does a lot of things at a cellular level, but the fundamental key is that it interferes with the ability of DNA to replicate. And so it kind of locks on to the DNA molecule and stops the process of cellular division. One of the major impacts of Barney Rosenberg's work is kind of reflected in my own career. I started in my training before cisplatin was available, and so almost every cancer that was metastatic actually caused the death of patients within about 2 to 3 years, sometimes faster. That was particularly dramatic because people who get testicular cancer are aged less than 25, 30. So they're young people, and these guys would show up with advanced cancer and be dead within 2 or 3 years. I was in my 20s. You look at someone in his or her 20s and you kind of think about all the parts of their life that they will lose. Once cisplatin became available, the expectation was to cure most of them. So it was a game changer.
I have the greatest respect for Rosenberg, a brilliant man. Cisplatin compounds effective against solid tumors all over the world, basically.
He got a lot of letters from patients that said, I owe you my life. And that was quite moving for him.
The work that we've been talking about here has saved lives at an incalculable level.
If he had been a cancer researcher, maybe he would not have been doing the experiments he had been, or wouldn't have considered the properties of platinum because there was an accepted wisdom about it.
It was an incredible discovery.